recording for this session. Okay, and it's 7 o'clock, so we'll let everybody get logged on and then we'll get started. So, hi everyone, welcome to the study session. So, the purpose of this study session is basically just to make sure that you're good and prepped for exam 1, which is going to open this Friday, right? And you have the entire weekend to take it. And then on Monday, we'll have a group exam. And there will be more details on like how your specific section is doing that, probably tomorrow and Friday. So we'll definitely talk about it more. But basically, to create this Kahoot, I did go through exam 1. So I have access to the exam. I can see all of the content that's covered. I went through the notes and I basically created simulated exam questions. And you'll have 20 seconds to answer each one. And I think there are a total of 35. So we'll go through those and we'll pause after each question, kind of talk about why that was the right answer. And then if there's any confusion or questions you have, feel free to ask those as well. So is everybody logged on to this? See, we have 2 people online. Let me see that. Okay, according to my count, everybody's on there. So we're going to go ahead and get started. So I'm assuming you know how to play Kahoot. So when the question and answers come up, you try to click it as fast as possible. Okay, yes, that is very good. So a living organism, a characteristic of living organisms is that they reproduce, they respond to environmental stimuli, and they consist of one or more cells, right? That's cell theory. So why has a heartbeat the correct answer in this case? Why is it not a characteristic of a living organism?
The plant would be one.
Right, a plant is a living organism, but does it have a heartbeat? Do bacteria have heartbeats? Yeah, so it's exception to the rule. What are some other characteristics of living organisms that you all remember?
Metabolizes, responds to stimuli, grows, reproduces, has more than one cell,
Yeah,
requires energy.
yeah, so yeah, definitely remember that entire list. That's going to serve you well. Because there may be a question that's similar to this and you have to pick out which one is not a characteristic. So very good. Alrighty. Yeah, and the faster you are, the more points you get, in addition to being correct. Okay. A blank is defined as members of the same species living in the same area. Okay. So we're kind of split on this one, and I get that the definitions are very similar, so it can be confusing. But this is going to be a critical concept to get on the exam because 100% you will be asked the difference. So you need to know. So members of the same species living in the same area, that is a population. So all of the gray squirrels on UC campus, that's a population. Community is one step up in our hierarchy. If we remember, like the largest is biosphere, the smallest is atoms, and I would definitely kind of note that level of organization. But for community, it's members of different species living in the same area. So all of the students and staff on campus, the gray squirrels, the geese, the trees, that is a community. Okay, so are we clear on the difference? Because I know there was kind of like a 50-50 split. And then an ecosystem, I noticed somebody also selected this one. Who can tell me what an ecosystem is?
That is a combination of living and non-living organisms.
Yeah, you're absolutely right. So it's a combination of all of the living and non-living things. So, you know, all of the animals, all of the plants, but also, you know, the temperature, the pH, the soil, the rocks, stuff like that. Very good. And nobody selected organisms. Oh, we got a rearrangement of the board. Okay, a blank is defined as members of the different species living in the same area. Okay, very good. And then one person selected ecosystem. The thing with this game is that it's not like 100% accurate because some people are trying to get it so quick that they automatically select something instead of the other. I'm assuming that this was the case, but remember, community, members of different species living in the same area, population, members of the same species. And then an ecosystem is the next step up. It's even larger than a community because it's all the living and the non-living things. Okay. Ooh, Jack is on fire. Okay, true or false? Deductive reasoning is based on observations and results in a broad conclusion. For one person, okay. So, this is exactly a 50-50 split. So, who wants to tell me why the correct answer is false?
Because deductive is like a ton of evidence resulting into one conclusion. So like if you want to relate it to like a crime, a detective is trying to figure out all the evidence and getting everything to result to one thing, so like who the murderer is or who did the crime. And inductive is like the study of what, say like this lake has, it's something that can be true over time, like when it's inductive.
Yeah. Okay. Does anybody else have anything they want to add to that, or? Okay. Yeah, so those are all really good points. But what really helps me is looking at the end conclusion. Like you said, with deductive, it's a single instance. It's very specific. While our inductive is very broad and encompasses many things. So what would make this statement true then?
Results in a simplified or certain conclusion.
Yep. So we change that.
Or if you just change inductive.
Yes. So 2 ways. No, you think in different ways, you know, but both of those are true. Very good. Okay, so let's keep that in mind, because you will 100% either have to get the definition of it, or she'll present you with an example, like Harold is bald. Harold is a grandfather. All grandfathers are bald. Inductive or deductive. And you'll have to select. And it is multiple choice, remember, so the correct answer is there. You just have to use your skills and root them out and figure out which one's the right one. Great. Ooh, Ian is on fire. And Angelina has moved up. Okay, I'm determining the effects of fertilizer on the height of corn plants. What is the independent variable? Excellent. Okay, so I heard from the grapevine that a lot of people were struggling with independent and dependent variables, but from the looks of this, it looks like it's not you all. So great. But just remember that our independent variable is usually the variable that's directly manipulated by the researcher, and it causes usually direct change in our dependent variable. That's why the dependent variable is called dependent, because it's dependent on the independent. Making those connections. Very good. Also rearrangement. Okay. Same thing, but what is the dependent variable in this case? very good. Plant height. So like I just mentioned, our dependent variable is dependent upon the independent variable. So our independent variable is fertilizer. So we're looking at the effects of fertilizer on corn, the height of corn plants. And our dependent variable is usually the variable that is directly measured by our researcher. And, okay, so this one right here, presence of fertilizer. Remember in the previous question, that was our independent variable. So they're either one or the other, they're usually not both. Is that clear, the difference between independent and dependent variables? Because that is not only important for lecture, but it's also really important for lab. You will have like a lab midterm and then a lab final, and this will most definitely be on that. Difference is clear? Everybody's doing good? Okay. If not, stop me, be like, Fallon, I do not understand this. Please explain it in a different way or something to that degree. Let's go ahead and turn the volume into it. Okay. Okay. So, same experiment, I decided to give each plant the same amount of soil, sunlight, and water. What is that called? I'm keeping some things the same across the board. Okay, so kind of split 50-50. So, in lecture, well actually, depends on which section that you were. In my section, we revisited this in lecture, but most of you only talked about it in lab. So the difference between a controlled experiment and a control group. So your controlled experiment is basically your baseline. So you're keeping some things the same for all of your groups, so you can say, okay, this variable directly causes change in this variable. It's a way to kind of check yourself. So in my experiment, if I'm giving each plant the same amount of soil, sunlight, and water, and all of those things, and the only thing I'm varying is that fertilizer, then that's a controlled experiment, keeping everything the same. Yeah. Okay. And then a control group, remember, so we always, typically in an experiment, we have a control group and a treatment or an experimental group. So in this case, I would have like a group maybe that doesn't receive fertilizer, and then one that does. The one that doesn't receive anything is my control group. And CJ, we'll give you a chance to log on, okay?
Okay, thank you.
No rush. I can give you the game pin because it's not popping up right there. Okay, but I'll take this moment to see how everybody's doing online. I noticed that we have a new person. Are you able to sign into the Kahoot? Just tell me when you're ready for the pin.
Okay, I'm just back to Wi-Fi.
Okay, great. Okay, it is 1099162. Is this clear though? The difference between a controlled experiment, like those baseline conditions, and then a control group. So you're able to get in?
Yep.
Okay, perfect. So we'll move on to the next one. Okay, true or false? For the fertilizer experiment, the control group is the group that doesn't receive the fertilizer or the treatment. Okay, very good. So remember, we just talked about it. So the control group typically is your group that doesn't receive your treatment or whatever you're investigating. So in this case, it would be the corn plants that don't receive the fertilizer. Our experimental group is the one that receives the fertilizer or the treatment, whatever we're investigating at that moment in time. It looks like majority of you got that, so good job. Ooh, Tommy's on fire. True or false? A hypothesis is an educated prediction that must be testable and falsifiable.
I'm being sabotaged.
Oh no. Okay, we'll see if
It just let me focus. Now I'm at a good place. I think somebody's sabotaging right
Might be me.
oh, it happens.
It's okay.
Okay, so good job. Looks like most people got this right. So a hypothesis is an educated prediction, right, or educated guess based off of previous observations and tests that we've conducted. And it also must be testable and falsifiable. Testable in that, we can test it. Usually the example that Dr. Nell likes to give is the flying spaghetti monster. If I hypothesize that there's a flying spaghetti monster out in space somewhere, that's not a good hypothesis because you can't prove that. And you also can't disprove that. And that's a part of falsifiable. Your hypothesis must at least have a chance that you can test it and it'd be false. It's like a 50-50. So yeah. Definitely remember that a hypothesis must be testable and falsifiable. 100%. That will pop up. Yeah. Oh. It happens to the best of us. Okay, a blank consists of generalized statements that are based on repeated observations and events and used to predict future events. Okay, so why is law the correct answer in this?
I thought I was right.
I thought theory was a bunch of them or something.
It is.
Okay.
So theory is very broad. That generalizes maybe where you're like, okay, that's theory. But what gets you right here, used to predict future events. A law can do that because it's typically mathematical and it happens literally every single time. Like, for example, I take this piece of paper and I let go of it with glass.
A fall.
That's Newton's law of gravity. No matter where I'm at on Earth, it's always going to happen like that.
Is there like a specific reason, like a specific difference between the two so I can remember, like, how to work this? Like, yeah.
Okay.
Good way to remember this.
Okay. Let's see. That's usually how I do it. Like the definition, so generalized statements based on repeated observation of events used to predict future events, typically mathematical in nature, like Newton's law. One of his laws is force is equal to mass times acceleration. It's mathematical, like it always happens.
Okay.
But a theory is actually more broad in nature, and it includes multiple hypotheses. It actually includes some laws too,
Okay.
to make like a big thing. So like cell theory that we're talking about, it has some math involved in it. Like we talked about the surface area to volume ratio, like all that math that goes into it, the upper limit and lower limit. And then it has a much, like some idealized things too.
Okay, cool.
So it's like the broadest. Does that make sense? Okay. Cool, so we definitely need to be clear on those definitions and the differences between the two. So hypothesis is the smallest. Build on that a little bit. Throw some math in there. A law. We take hypotheses and laws together. Theory. Theory is the largest. Okay. Does that make sense to everybody?
Yeah, that really works.
And then if it didn't, just picture me throwing a paper ball across the room on test day. Newton's law.
Keep me out forever.
A blank attempts to broadly explain and collect groups of laws and observations. Okay, so very good. So there we see it again. We have, it's the broadest umbrella. It includes everything. And, okay, hypothesis. So I see there are a few people who selected that. So remember, our hypothesis is just an educated prediction based on observations or tests. It doesn't have anything really to do with those laws. Now, we like to use hypotheses to create laws, but it doesn't work the other way, typically. Is that clear why a hypothesis was not the correct? Because it didn't accurately describe what was going on there.
So the questions on the exam are going to be worded like this?
Very similar. Uh-oh, CJ has a streak. Don't want to brag or anything, but select the correct monomer polymer pair. Okay. So, there's a little bit of confusion, but that's okay, we can clear that up. So, what do I mean when I say monomer? What?
Single unit.
Single unit, right? So then a polymer is
Multiple units.
multiple units, right? So basically pairing the correct one. So the first one, or red, why is that wrong? So I've paired an amino acid and a polysaccharide together. Why is that fundamentally wrong?
Amino acids create proteins and polysaccharides are carbohydrates.
Very good, right? So that's good. And then so the same thing with this one, right? Because another name for a protein is a polypeptide. That one's out. And then our nucleotide and proteins, what are nucleotides a part of? Very good, right?
Quick question. You said to match it, and amino acid is, you're saying it is a monomer polymer, it is a polysaccharide or poly.
So some of these may be a little bit messed up. And that was to guide you to the right answer, because on the exam, they will be messed up too. So this is the correct answer, because an amino acid is a monomer, right? And it comes together, many of those come together to form a polymer, which is the protein or the polypeptide.
Okay.
Now, some of these aren't exactly monomers or polymers, but that requires you to know, okay, what are monomers and what are polymers. And that's what you'll have to do on the exam. So is that clear? Like how we kind of figured that out together? All right, what is the function of carbohydrates in the body? This is an in-class assignment for Very good. So carbohydrates are your fuel or your energy for the cell. And I think in a couple of units we'll talk about cellular respiration and exactly how carbohydrates are broken down to fuel the cell. Very good. So all of these other functions, what are these for? Like enzymes. Which biomolecule? Protein. Transport?
Also proteins.
Proteins, right? What about storage?
Lipids.
Very good. And like we say in class, actually if there is a function that's needed in the cell, proteins probably do it. They're so diverse. Oh, and 3 players hit answer streak 3. Good job. What type of biomolecule is this? Okay. We were a little mixed, but mostly carbohydrates, so that's good. So we also selected protein and nucleic acid. Let's keep going. Oh, it's horrible. Okay. Let me go back. Okay. So if we visualize in our head what was going on, what was the first thing that you noticed when you looked at that structure?
It has carbon and hydrogen.
Right, and you said ring structure, carbon, hydrogen, hydroxyl groups, like a ton of them, those OH groups. That should be your signal that that's a carbohydrate. Now, we also selected proteins and nucleic acids. So with a protein, what groups would I be looking for?
Amino.
Amino group?
The N one. I don't know. Sorry.
No, you're good. No carboxyl. But remember, our lipids also have carboxyl groups, right? So that amino group that you mentioned, that's actually a really good thing to look for because carbohydrates don't have it, lipids don't have it, nucleic acids, because it's an amino group and those are amino acids.
What's the amino group? I don't know.
So it's the nitrogen and the two hydrogens.
All of it's nitrogen based.
Hmm?
Okay.
So would you just suggest like all those diagrams to learn them, just like flashcards or Quizlet or something? It's really hard for me to picture the diagrams and put them with the name or like a...
Yeah. So I actually think that Quizlet would be a great idea. And just knowing how to pick out those iconic traits. So like, even if you didn't know what a carbohydrate looked like you didn't know like a ring structure or anything. Exactly. It's in the name, right? Carbohydrate. That sort of thing. Or looking at the many hydroxyl groups, or like for a lipid, if it's literally just like a long carbon chain with nothing but hydrogens, that's probably your signal. It's non-polar, that's a lipid. Just stuff like that. But yeah, and use the pictures in your notes. Like maybe just like show yourself a picture of it and just be like, okay, what biomolecule is this? And just kind of try to replay it back to yourself. And then nucleic acids. So we remember that nucleic acids are made of nucleotides, right? So who can tell me what's in a nucleotide? Phosphate group, very good. There are three components. There's a phosphate group, a sugar, right? 5-carbon sugar, and then a nitrogenous base, so your ATGC or your AUGC. So nucleic acid, like a component to it, would look a lot more complicated than the structure. And I think the carbohydrate that I just showed you was glucose, actually. Hey, the person on the door. Okay, another one of these questions. What type of biomolecule is this? I know it looks scary, but just remember what we just talked about, iconic features. Okay, so we're split. So usually when you're given a question and you're asked what type is this, not or none of the above is usually not the right answer. That's true for all classes that I've been in, just FYI. Okay, so lipid was the correct answer. Nucleic acid is protein. Okay, so who can tell me why lipid was the correct answer?
The lack of oxygen.
Lack of oxygen, right? I hate that it doesn't pop up correctly, but these 4 fused rings, do you remember that from lipids? What was that characteristic of particularly? Do you remember?
Steroids, right?
Steroids, right? And those are a type of lipid. But even if you didn't remember that, like you said, the lack of oxygen. So in biochemistry and organic chemistry, you'll learn more about this, but you learn basically in here that every point is a carbon, and those carbons are surrounded by hydrogen. So what you should get from looking at this structure is that this is mostly carbon and hydrogen. And when things are made up of mostly carbon and hydrogen, they're non-polar and they're hydrophobic. and that's characteristic of lipids, right? So that's kind of how we work through that. And then for nucleic acids, remember we talked about we would see a phosphate group, so a phosphorus connected to 4 oxygens. We would see a carbohydrate, a 5-carbon sugar, and we would also see a nitrogenous base. And we didn't have all those components present, so that's why it's not a nucleic acid. And then for a protein, remember we need to see that amino group in particular, in addition to that R group, which may be different depending on the amino acid, and also carboxyl group somewhere. Okay, so does that make sense? Kind of how I work through that?
Yes.
That is a favorite question, by the way. Put a steroid on there, ask you what kind of biomolecule it is, and you should be able to work through and be like, that's a lipid, mostly carbon and hydrogen. 8 correct answers in a row. You go, CJ.
Thanks.
And look at our board. Good job to everybody who's on here. Okay. Which of the following processes is demonstrated below? So in this photo right here. Okay. So what I noticed here is that nobody clicked linking or ionic bonding. That's good. So we realized that hydrolysis and dehydration synthesis are the 2 big processes that we need to know. So who can tell me why it was dehydration synthesis?
Because you're breaking them up and then you're taking, well, okay, the main thing I'll look at the end, you have to get the H2O at the end. You know what I'm saying? That's the big thing. And then you got the oxygen, the same bond right there.
Right.
So we just gotta look at that.
So
Yeah.
hydrolysis is the one that's removing water?
No, this one right here. So dehydration synthesis means dehydrate, we take away water, and we synthesize, create something bigger. So here are 2 monosaccharides that we are taking the water out, and that's why it moves over here, where's the leg. So we take the water out, and then they're linked at the oxygen, forming the covalent bonds. Does that make sense? No? Okay. So what's confusing about it?
Because you have water there, so how are you taking it out? You don't, so that it's not there in the beginning at all, right?
No.
Okay.
So do you see how these are red?
Mm-hmm.
Taking it out is right over here. When you write a chemical equation, this is more of a chemistry thing. If it's being taken out over here, it's going to appear over here. This is like a byproduct.
Okay, gotcha.
So it's just like a logistical thing.
Okay.
So what you really need to focus on is, are we creating something bigger, or are we breaking up something that's big into smaller units?
Okay.
And if we're making something bigger, dehydration synthesis, if we were going this way, so we were adding water to this and breaking it up into 2 monosaccharides, that is hydrolysis, because we're adding water to break things up. Okay. Okay. So this is probably going to be the opposite one. I apologize, this is so small. Okay, yes, very good. So that is hydrolysis, and why it is hydrolysis. So we have our polypeptide, right? And this just shows you a component of it. So we're adding water and we're breaking it up into our amino acids. So that process right there is hydrolysis, when we add water to something and we're breaking it up into smaller components. So just make sure that you really understand the difference between the 2, and there are biomolecule practice exercises. I know we keep preaching those and pushing those on you, but I promise you they're a huge help, especially if you struggle with these 2 concepts. The biomolecule practice exercises, I think they're under week 2 and week 3 on like both e-learn pages. So You can go on there, it's a PDF, and you just click on it and you can go through them and work on them.
Like it's like a link.
Yes. So you click on it, it's like a document that pulls up.
Okay.
Okay. Uh-oh. Answer street. Okay, which functional group is not found in the given structure? Okay.
I really hated that one.
We're split on this one a little bit. Okay, this pops up. Okay, so this is a picture of what? What is that?
Protein, right?
So a component of a protein, right? Amino acid. Very good. Okay, so with an amino acid, we already talked about some of these groups, but now we're going to identify them. So this nitrogen with two hydrogens, this group is the amino group, right? Okay. This group right here, this carbon double bonded to this oxygen and this OH group, this whole thing is called carboxyl. So if you have trouble like dealing with that one, because I know we talk about carboxyl and carbonyl. Carbonyl is seen with carbohydrates, and it's basically a carbon double bonded to an oxygen, but instead of an OH, it's just a hydrogen. Carboxyl is this group. And I use that to, how I remember that is an OH group is a hydroxyl. This is basically carbo, so carboxyl, put them together. And then this one right here, this is our variable residue or our R group. So it depends. So the only one that we don't have here is our carbonyl, right? Because that's seen with carbohydrates. And this is an amino acid that makes up a protein.
Can you explain what variable residue is? I guess I didn't think it was something that.
Okay. So usually, I know that we talked about like variable residues in our section, but Dr. Arnett may have just said like the R group. And that's
Yeah.
basically what I meant by that. So it's this one right here. Every amino acid has a different R group, and you learn that it can introduce like a positive or negative charge. It can make it polar or non-polar. And the R group is basically just what generates the diversity. So like the 20 different amino acids. But if you see it on the exam, it'll probably be R group, just to make sure everybody knows what we're talking about. Ooh, another person on the board. Okay, all of the following describe carbohydrates except pay attention to those keywords on the exam as well. So we kind of touched on this briefly, but remember with our carbohydrates, an iconic characteristic is that they have a lot of hydroxyl groups. And because they have a lot of hydroxyl groups or a lot of oxygen, that's going to make them polar. And things that are polar are automatically hydrophilic because water is polar. And we know like dissolves like. So that's kind of how you connect all of those in together. What kind of biomolecule is typically hydrophobic or nonpolar? Lipids, right? So they're like in direct contrast to one another. Does that make sense?
Yeah.
That's how we connect all those together. All of the following describe lipids except
Protein.
yeah. So you just mentioned active enzymes. Remember, only proteins, those biomolecules are able to do that. But I kind of put this one in here to do a little bit of a trick. Because there is a unique type of lipid that has polar and non-polar components. Does anybody remember what that is called? Yes, very good. Phospholipids. And those are like probably one of the most important ones because we're talking about the cell and the cell membrane. We're going to be talking about that on Wednesday. All of the cell membranes of all cells are made up of phospholipids. That is crucial. And phospholipids have hydrophilic heads and hydrophobic tails, right? So that's where that comes from. And we also just said that they're nonpolar and hydrophobic because of those long hydrocarbon chains they have and virtually no oxygen or very little of it. Very good. Oh, Haley. Is an answer true? Okay, true or false? Primary structure of a protein is the linear amino acid sequence held together by peptide bonds. Okay, good. So this is also going to be a major thing on the exam, knowing all of those structures of the protein, so the primary, secondary, tertiary, and quaternary, knowing what they look like and what kind of bonds or interactions are holding them together. So the primary structure of a protein is the linear amino acid sequence, and it's held together by peptide bonds. So it's nothing special. So if you were in Dr. J's class, basically just the phone cord stretched out. I know Dr. Annette likes to do some type of origami folding exercise, so if you can just visualize that in your head as you go through. Yeah, it's just basically like a definition thing, knowing the characteristics. Uh-oh, CJ is making us come back. Okay, name the protein structure. Alpha helices and beta pleated sheets held together by hydrogen bonds. Is that primary, secondary, tertiary, or quaternary? Okay. So we just talked about primary, right? And how that's just a linear, nothing special about it. Amino acid sequence held together by peptide bonds. So that's why that's not the answer. So secondary is the correct answer because that's where we see the alpha helices and beta pleated sheets. So who can tell me why tertiary or quaternary aren't the right answers? What do we see with tertiary? 3D structures, very good, right? And what are those 3D structures typically held together by?
Was it polypeptides, or was it?
So, yeah, like a protein is a polypeptide.
Yeah.
But like our three-dimensional structure is held together by some kind of interactions. Side chain, very good. And then quaternary, what's the difference between quaternary and tertiary?
Isn't like all mixed up together?
Two come together.
Right. So not all proteins actually reach the quaternary stage because for quaternary we have multiple proteins that come together and form like a big protein. And those are held together by hydrogen or ionic bonds. And those might just pop up in later questions while we have a rearrangement. Okay, name the protein structure. 3D structure held together by side chain interaction. Okay, so we just talked about this as tertiary structure. It's 3D held together by side chain interactions. I think there's a question on quaternary, so I'm just going to save that for the next one. Which of the following are side chain interactions? Okay, So we said that the tertiary structure of a protein is held together by side chain interactions, but it's crucial that you understand, okay, what does that mean? What are those? So those can actually be hydrogen bonds. They can be disulfide bridges, so the connections between sulfur, which is another element, and then hydrophobic interactions. So nonpolar groups repelling one another, essentially. And then, well, actually coming together, because like dissolves like, right? Like things want to be with like. Excuse me for that. And then ionic bonds are another type of thing that you may see as well. But just knowing like what some of those interactions are. Cool. Megan has an answer. True or false? Proteins are functional once they reach their tertiary structure. Okay. So, this is true, right? Because their primary structure, not functional. Secondary structure, not functional. Tertiary, we need that 3D structure. We need to be folded up just right because remember, structure determines function. And if you mess up the structure of a protein through like denaturation, if you mess with the pH, you mess with the temperature, proteins will unfold and they lose their function because structure is everything. So that is true. Okay. Name the protein structure. More than one polypeptide held together by hydrogen or ionic bond. So definitely study these, commit them to memory. It's going to make your life a whole lot easier, I promise. So more than one polypeptide, more than one protein coming together, forming hydrogen or ionic bonds. Remember, that's our quaternary. Like our hemoglobin, for example, that requires 4 like proteins to come together and make one big one. And not all proteins reach that level, just some have it. Okay. True or false? A nucleotide is a monomer of nucleic acids. It is made of a sugar, phosphate group, and a nitrogenous base. Okay. So remember, a monomer is a single unit, and when you put it together, it should form a polymer. Nucleotides, many nucleotides come together to make up nucleic acids, or RNA or DNA. That is true. What is also true is the components of a nucleotide. Remember we said that 5-carbon sugar, that phosphate group, and the nitrogenous base, ATGC if it's DNA, AUGC if it's RNA. That is true. That makes up the nucleotide. Jonathan has an answer streak. True or false? There are 2 nucleic acids, DNA and RNA. Very good, yes. There are 2, only 2, DNA and RNA. Ooh, Sophia's making a comeback. Okay, what is the major difference between RNA and DNA then? Okay. So, good, most people got it. DNA is single-stranded. Why is that not true? Double-stranded, right? RNA is double-stranded. That's single-stranded, right? So I mixed those up purposefully. Okay, RNA is the only nucleic acid. In a previous question, we said there are 2. That's not true. So then DNA is used for long-term storage of genetic information. What is RNA used for? Very good. And I feel like this is more easy to tie in because we'll eventually talk about DNA replication, go through the specifics of that. And we'll also talk about transcription and translation, and that's how we make proteins in the cell. Okay. A few more questions and I'll let you go. Oh, Lily is back with an answer streak. All right, which of the following is a function of protein? Okay. So remember we said that there's a function and the cell needs it, the protein probably does it. They do function as enzymes. They do transport. We'll talk about transport proteins and specifics of that soon. Protection, regulation. They do a bunch of different things in the cell, and I would definitely memorize a few of those functions, but all of these are functions of proteins. So be wary of that on a test, because enzymes is a correct answer, but it wasn't the best answer. So a lot of times that'll get you, so make sure you carefully read what's going on and select. What type of bond is formed between monosaccharides during dehydration synthesis? Okay, good. Most people selected covalent. So that's that carbon and that oxygen bond sharing the electrons. Ionic is typically when they have different charges. So like your sodium, which is positively charged, and your chloride, which is negatively charged, coming together to make NaCl, which is table salt. That would be an ionic bond. Hydrophobic interactions, remember those are associated with the side chain interactions. So that's not even really a bond. And then peptide bond, what are these associated with?
Amino acids.
Yeah, amino acids linking those together to form those proteins. Very good. So covalent bond is what's formed. Okay, true or false? Phospholipids have polar or hydrophilic heads and nonpolar or hydrophobic tails that allow them to form a lipid bilayer. Very good. So remember we talked about in previous questions about how phospholipids are a unique type of lipid because they have polar and nonpolar components, and those allow them to form that lipid bilayer. Good job, Lauren. Okay, phospholipids form a lipid bilayer that forms the cell membrane of all cells. True or false? Okay, very good. Yes, that's true. Now remember we talked about how the hydrophilic heads and hydrophobic tails allow them to orient themselves to form a lipid bilayer, and the whole purpose of that is so we can form that cell membrane that's surrounding all of our cells, prokaryotic or eukaryotic. And we'll talk more about those probably in class tomorrow. Okay, good job, Ian. 2 more questions. The blank is best described as the minimum volume that cells require to house all of the essential organelles and material. Okay, very good. So I remember when I was in Bio 130 and we started going over cell theory and they went over the lower limit and the upper limit and I was like, oh my God, what is this? Like, what are they talking about? Like, I just completely glazed over. So I want to include some stuff about this because it probably will pop up on the exam. So remember the lower limit is basically the smallest that the cell can possibly be to hold all of its organelles, you know, those many organs we'll talk about that perform essential functions for the cell. So that's our lower limit. And that is a part of cell theory, that doesn't match up with the definition. So that's why cell theory is not the correct answer. Okay. Final question. The blank is best described as the maximum surface area to volume ratio that will allow for efficient nutrient and waste exchange. So lower limit was the previous one, upper limit then is this one. This is basically the largest that the cell can get before, you know, its nutrient requirement is too large and it generates too much waste to efficiently get out of the cell. Good. So you all are troopers. I hope that helps you understand stuff a little better. Maybe some like test taking skills. Good job to everyone, but especially good job to our leaderboard. Congratulations. So, are there any questions or concerns, anything like that? So I think there's like 26 maybe, but I'll double check and we'll send email
Are they all
about
multiple choice?
it. They're all multiple choice, no essays for answer, anything like that.
How can we get the cookies?
I'm going to send it out to everybody if you put your name down. I'm going to send it out and I'll leave it open so you can play it as many times as you want to. And if you have any questions, please feel free to email me. It was nice to see all of you and I hope this helped.
Yes, it is.
Good. Okay, is everybody doing okay online?
Thank you for your help.
No problem at all. You'll have a good night.
Dude.